In the time that it will take me to give this talk, your immune system will, in all likelihood, be fighting off a couple of infections. It will be dealing with countless damaged cells in your body that need to be disposed of. And it will also be actively preventing cancer. And you won't even notice. So I am Caetano Reis de Souza. I am a Principal Group Leader and Assistant Research Director here at the CRIC, as was mentioned, where I lead the Immunobiology Laboratory. And this is a group of outstanding researchers, you can see them on this photo, who are dedicated to studying and understanding the immune system. And so today we're going to discuss a little bit about the immune system, and we're going to discuss first what is immunology or immunobiology. And it's really very simple. It is the study at its very basic of the system that allows our bodies to protect themselves from disease. And we know that disease burden and disease mortality will be much, much higher if we did not have an immune system. And of course, the immune system is also the system that is elicited by vaccination, as we will mention a little bit later. And so it is perhaps easiest to think of the immune system as a very simple circuit. You have an insult, it is detected, and it puts in place a response that is designed to neutralize that insult. And the insults that we are dealing with when we're talking about the immune system are infection, cancer, and also damage to tissues or tissue malfunction. And so the way that the system works is by using cells. Cells of the immune system are white blood cells. And we tend not to think of the immune system as an organ, but of course, if you were to put all of these white blood cells together in one place, they would occupy a mass about the size of that of the brain. So it's quite a sizable organ, if you will, albeit one that is dispersed throughout the body. And these cells are in constant communication with one another, helping coordinate the immune response. And of course, that involves molecules that allow for the communication, and molecules that allow for the cells to perform the functions that they have to perform. Now, on the left-hand side of this slide are the good things that our immune system does. It defends us from infection by viruses, by bacteria, fungi, protozoa. These tend to be small microorganisms, but the immune system can also deal with big things, worms, ectoparasites, and of course, as mentioned, it can deal with cancer. Now, on the right-hand side are some of the detrimental effects that the immune system can have. Dysregulation of the immune system can lead to severe diseases such as autoimmunity, also allergy, and for transplant recipients, it can lead to graft rejection. Now today, we are going to mostly focus on the left-hand side, on the defense aspect, but it is important to realize that this is a careful balancing act, and we will mention in passing some of the regulatory mechanisms that ensure that this equilibrium is maintained, whereby we can achieve defense without the detrimental effects that could bring about. So let's start with a brief history of immunology, and I'm going to take you all the way back to ancient Greece, 430 BC, when there was a massive plague called the Plague of Athens. It actually wiped out 25% of the Athenian population, and it was probably not the plague as in the one that we had in Europe in the Middle Ages, the bubonic plague. It was called a plague, but it was likely to have been something like typhoid fever. And the key figure that I want to mention is Thucydides. He was a historian who actually survived the plague. And he is probably the first person to ever have described the state of immunity. And this is what he wrote, yet it was with those who had recovered from the disease that the sick and the dying found most compassion. For the same man was never attacked twice, never at least fatally. And effectively what this was is a description of the fact that if you were fortunate enough to recover from this plague, you were immune to subsequent attack. Now the word immune and immunity does not come from the Greek, it comes from the Latin. The word immunis meant exempt from taxes. Now, as an immunologist, I have tried to use that argument with the tax authorities. I have not had much traction. And so the next bit of our story actually takes us forward all the way from antiquity to 18th century England. And it has to do with a devastating disease called smallpox. Now, smallpox has caused devastating epidemics throughout human history. In the 18th century alone, it was killing about 400,000 Europeans a year. And it was characterized by these pus-filled bubbles that appeared all over your body. And if you weren't killed, you were effectively disfigured for life. You were pockmarked, as it were. Now, one particular individual, Edward Jenner, a physician scientist, he has a statue of Edward Jenner at Kensington Gardens in London. He actually made an interesting observation. Now, there was another disease called cowpox, a disease of cattle, that could be transmitted to humans, and it was often found in milkmaids. Milkmaids got cowpox because they were handling cows. And this also caused these pustules, but primarily in the bits that came into contact with the cows, i.e. the hands, and didn't cause this spreading disease. And also, unlike smallpox, it was not that serious or fatal. But what was noticeable is that milkmaids very seldom had these pock marks. In fact, they were famed for their beauty, which basically meant that they were not pockmarked. And the question that was being posed by Edward Jenner is, why is it that milkmaids catch this cowpox but seem to be immune from catching smallpox? And so the hypothesis, he was a scientist, so he formulated a hypothesis, and the hypothesis was that maybe these milkmaids did not catch smallpox because they had caught the cowpox previously. And so he proceeded to do an experiment, and this is the experiment that he did. He took an 8-year-old boy, James Phipps, the son of his gardener, and he deliberately infected this boy with cowpox, pus taken from the pustules of Sarah Nelms, a milkmaid. The boy got a bit sick, he got a fever, but he rapidly recovered. And then he went on to do the second part of the experiment, where he now deliberately inoculated the boy with smallpox. And fortunately, the boy survived. And he went on to do this experiment with an additional 23 volunteers. And so we can question the ethics of this experiment. But the point was that this was effectively the first demonstration that you could actually vaccinate individuals against a severe disease, in this case by giving them a mild disease. And of course, the word vaccination again comes from the Latin vacca, or cow. In fact, the cow from which Sarah Nelms had caught her cowpox was called Blossom. This is the hide of Blossom at a museum here in London. And I think we can only be fortunate that they didn't decide to call it blossomization. Now, of course, this was then rolled out as a vaccine against smallpox. And in fact, vaccination against smallpox has been extraordinarily successful. Smallpox has been eradicated from the planet. It does not exist anymore. But naturally, people were a little bit worried about vaccination This is the sort of 18th century version of the fear of vaccination. You can see in this cartoon Jenner vaccinating patients against smallpox. On the right-hand side of the cartoon, you can see the fear that people would have bits of cows growing out of their body. And this fear of vaccination, of course, persists to this day. Now, this, however, is an unfounded fear because we know vaccines are extraordinarily safe. And importantly, this is a study that was just published last year that looks at the outcomes of childhood vaccination in the last 50 years. And the take-home message is very simple. Every hour, around 360 children are saved thanks to vaccines. So vaccination is actually the most, or one of the most successful interventions ever developed by mankind to fight disease. It has eradicated smallpox from the planet. It has nearly eradicated polio and massively decreased the incidence of many other diseases, including measles, mumps, rubella. And what vaccines effectively do is that they mimic or they induce that natural state of immunity that can occur if you develop disease and survive, such as Thucydides described in the Plague of Athens. And so let's look a little bit more at how the immune response actually occurs. And there are two fundamental branches to the immune system. One is called the innate immune system or inflammation. These terms are used interchangeably. It's a quick and general response, a first line of defense, if you will. And then there is another one, a slightly slower one, but it is more specific and has memory, which is called the adaptive immune system. And we will cover all of this in the next few minutes. So inflammation, again, we go all the way back to ancient Rome. Aulus Celsus first described the 4 cardinal signs of inflammation, which are used to this day to describe inflammation, and you will recognize them. If ever you've had a wound, you will recognize the fact that one of the first responses that you get is redness, heat, swelling, pain. And inflammation involves white blood cells, and the white blood cells that are critically involved in inflammation are called phagocytes. And these phagocytes were actually first described by Mechnikov at the end of the 19th century. And he noticed that there were some white blood cells that were able to gobble up particles, and he correctly surmised that this was a mechanism of defense. And you can see that in this cartoon on the left, you've got some bacteria, they've been gobbled up by a phagocyte, they end up in these vesicles inside the phagocyte where they are subsequently destroyed. And I'm going to show you a little video of a phagocyte. This is in a blood smear. The dark round cells are bystander red blood cells. You can ignore those. Focus on the amorphous shape in the middle. And then you've got some bacteria. Now, these bacteria are moving around through Brownian motion, just the motion of the liquid. But notice that phagocyte knows exactly where the bacteria are. It's following them around, pushing away through the red blood cells. Every time the bacteria moves, it turns until eventually it is able to catch up with the bacteria, gobble them up for destruction. So this is a very precise and well-defined response that allows for the elimination of some bugs that might infect you. But this phagocyte is not just gobbling up the bacteria and destroying them, it is also sending out signals that signify that it has encountered the bacteria, and these signals are then responsible for recruiting additional phagocytes from the bloodstream into the affected tissue, such that you now have a whole plethora of cells that can contain the infection, and in some cases that leads to the elimination and resolution of that insult. And it wasn't just doctors and scientists who became aware of phagocytes when Mechnikov described them. In fact, shortly thereafter, this became quite widely known. And here's an excerpt from a play by George Bernard Shaw called The Doctor's Dilemma, in which one of the characters, Bibi, actually says there is at bottom only one genuinely scientific treatment for all diseases, and that is to stimulate the phagocytes. Stimulate the phagocytes, drugs are a delusion. So how do you stimulate the phagocytes? And microbes obviously are the things that the phagocytes can target, and microbes are also the things that often trigger these phagocytes. And the reason why they can trigger the responses of the phagocytes is that microbes have molecular signatures that differ from the molecular signatures of your own cells. So here's just an illustration, a cartoon of a yeast cell, a fungal cell, and you can see on the surface of this cell this little coating which is made up of all of those molecules. The names are on the right, the names don't really matter, but the point that I would like to make is that none of these molecules are made by your cells. And so phagocytes have developed receptors, as we call them, molecules that are able to bind to these molecular signatures of microbes. And of course, most of the time, these receptors are not going to be engaged by anything because there is no microbe around. But if they are engaged, it must signify the presence of a microbe. And that engagement can now trigger those receptors. It can cause the cells to internalize the particle that has triggered them, as you can see in this video here, where you've got a phagocyte gobbling up the yeast cells. You see those red rings around particles that are being engulfed. The particles are the dark spots that you then see against the green. And so that is that engulfment reaction that you can see in a real cell down the microscope. And this is actually the signals that are then emanating from that encounter. You've got here the yeast cells have been engulfed. They are now inside the vesicles, inside the phagocyte, and that red cloud are these signals that are now being put out by the phagocyte to effectively ensure that this encounter is disseminated, or information about this encounter is disseminated and passed on to other cells. And so in some cases, as I alluded to, this reaction is sufficient to contain the infection, and you are none the wiser for it. But in many other cases, this is not sufficient, and we need another branch of the immune system to come into action, and that is this adaptive immunity branch. And there the key white blood cells are T cells and B cells. And so these are brought into play as a backup circuit to neutralize the insult. And T and B cells are absolutely critical for defense from infection. If you are unfortunate to have a genetic mutation that prevents you from producing functionally competent T and B cells, then you are extremely susceptible to infection. This is the unfortunate case of this boy, David Vetter, who became famous for having to live inside a plastic bubble so that he will be protected from the environment, protected from contact with others, and thereby avoid infection because he had no T and B cells. Nevertheless, regrettably, he was not able to contain infections and eventually succumbed to one. And so T and B cells are extremely important. They do slightly different things. So B cells are the ones that make antibodies. You will all have heard of antibodies. T cells come in different flavors. Some help B cells make antibodies. Others can actually specifically target infected cells and kill them, and thereby prevent the infection from spreading. Now, there's an important immunology word that I need to share with you, and that is an antigen. Again, you may have heard this word before, but an antigen is very simply a molecule that is capable of triggering a T or a B cell response. And so I'm going to represent antigens by this Joker card that you see here. And so I want to illustrate what happens if you encounter an antigen A, the Joker, and the first time you encounter it, you start to mount, in this illustration, an antibody response against A. It comes up, it peaks, and then if you clear that infection, then the response declines again a little bit, although it is now at a higher level than it started off with before you ever encounter this antigen. Now, the key thing is the second encounter. And you can see that if you re-encounter antigen A, you now mount a faster but also much more vigorous response. You get much higher levels of antibody. And this is what immunologists call memory as well as long-term immunity. But the other aspect of this is called specificity. And that is that this enhanced response only occurs for antigen A. If you come in with a second antigen B, the ace of spades, then you effectively mount a primary response against B at the same time as you might be mounting the secondary response against A. And so these two features, adaptive immune memory and specificity, are effectively the basis of immunity as described by Thucydides and of vaccination as discovered by Jenner. And so in the case of vaccination, for example, what you have is a vaccine that is an innocuous mechanism for making you encounter antigen A, so that you now have already mounted the primary response, such that if you were to encounter the infectious organism that contains antigen A subsequently, then you have this much more robust, much more rapid response, which is going to be much more likely to protect you. Now, I've illustrated all of this with antibodies, but very similar things happen to T cells. But let me just tell you a little bit about what antibodies do to bring this home. And so what we have is bacteria that are coming in and trying to invade your cells. They have these little feet and they're trying to stick to the cells of the body and they're trying to enter those cells. But along come the antibodies, these Y-shaped molecules. They can now bind to these little feet and thereby prevent those little feet from anchoring onto the cells. And now look at this. Here's a phagocyte and it comes along and whoop, it just gobbles up those bacteria that have been coated with antibody. And that illustrates another very important principle, is that I've talked about innate immunity and I've talked about adaptive immunity, but these work together. Innate immunity elicits adaptive immunity, but also adaptive immunity helps innate immunity function better. And so that is because actually these antibodies that are coating the bacteria make those bacteria more palatable to the phagocytes that engulf them more avidly. And in fact, in that play, The Doctor's Dilemma that I mentioned earlier by Bernard Shaw, one of the characters actually says, the phagocytes won't eat the microbes unless the microbes are nicely buttered for them. And so what the antibodies are the butter that makes those microbes more palatable. Okay, so now we know about the adaptive immune response. How is it actually induced? And so the key phagocyte that is effectively involved in inducing the adaptive immune response is called a dendritic cell. And dendritic cells are found in all tissues of your body, and they are there moving around a little bit and effectively acting as sentinels for any possible insult that might come into that tissue. Now, the T and the B cells are not in these tissues. The T and the B cells congregate in special tissues of the immune system that are dispersed throughout the body. There's a few illustrated here. We're going to talk about lymph nodes as an example. And I'm going to just give you a hypothetical example of an infection of the lungs with a virus. So this will be the example of a peripheral organ that's being infected. And what you have there are these dendritic cells, these sentinels. And the job of the dendritic cells is effectively to bring that antigen to the places where the T cells are and the B cells are, i.e. the lymph nodes, and that's what's depicted in this cartoon. So I'm going to focus on the T cell response for the purposes of illustration, but the B cell response will be very similar. So let's look at this in a little bit in more detail. Here are the lungs. The virus has come in, it's infected them, and the virus has got this viral antigen card, the joker, and the dendritic cell, the sentinel, is sitting around and effectively it now has some of these specific receptors for microbial signatures are now alerted to the presence of this virus, and that delivers a signal that tells the dendritic cell, stop being a sentinel, you've encountered something, it is time to take that something to where the T cells are in the lymph node. And this is what we call dendritic cell migration, which allows these cells to now arrive in lymph nodes and activate the T cells that are specific for that antigen. So this last bit is important, the T cells that are specific for that antigen. The way that the T cells work is that you've got lots and lots of T cells in your body, but each one of them is specific for a different antigen. In fact, it would appear that there's only about 1 in 100,000 T cells that might be specific for any one antigen. And so the job that you have to accomplish is to make sure that you find the right T cell, that 1 in 100,000 cell, that will be specific for that antigen. And so here we have a dendritic cell in the middle presenting this joker card, and you have all of these different T cells that are coming to audition to see if they are able to specifically recognize that antigen. And the red one does. That's great, but it's 1 in 100,000. So then what happens is something really remarkable. That one red cell proliferates furiously to now generate a whole army of T cells that have exactly the same specificity. And so now, instead of having 1 in 100,000 cells, you might have as many as 1 in 100 or 1 in 10. So you've now generated a whole army of T cells that are specific for that antigen. And by the way, this is also happening to B cells in parallel. And so, for example, if you've got a throat infection, you might say, oh, I've got swollen nodes in my neck, and you feel them. That's your lymph nodes that are expanding because all of these cells are proliferating to generate these large armies, which can now exit the lymph nodes. And in the case of these killer T cells, and in the case of the respiratory infection, they will go to the lung and specifically identify virus-infected cells because they are displaying that joker card, and they can kill those cells specifically while sparing the normal cells. And so what you have here is effectively a circuit where you've got dendritic cells as the sensors of bugs, the sensors of infection, the things that trigger the immune response and get the whole system going. And so we've got this extraordinary ability to detect bugs and trigger very powerful immune responses against them. Now, you may know of this book, Metamorphosis by Franz Kafka. Now, in this book, Gregor Samsa wakes up one morning and he's been transformed into a bug. And so poor Gregor does not know anymore whether he's a bug or a man. But you all know right? You all know that you're just as much bug as human, because you've got just as many bacterial cells in your body as you've got your own cells. And you've also got plenty of fungal cells and viruses. So I've just told you that we've got a whole system there that responds to the presence of bugs, and when it detects bugs, all hell breaks loose, and you get this powerful immune response to eliminate the bugs. But at the same time, I'm telling you, you're full of bugs. And the reason is that it's a little bit more sophisticated than I've made it sound. In fact, the immune system does something quite extraordinary. It discriminates good bugs, commensal organisms, from bad bugs or pathogens. And we don't fully understand how it actually does that. Now, part of the answer is that actually there is no such thing as an immune response. There are immune responses of different qualities, and some of them are designed to eliminate and others are designed to just keep things in check. That's part of the answer. But it is actually an area of immunology that we are still trying to investigate and trying to get a better understanding. How is it that we can coexist peacefully with all of these commensal organisms, and yet if we are infected, we can eliminate pathogens? Now, one of the answers has to do with localization. So here's your gut, and in your large intestine, you will have a large burden of commensal bacteria. So the diagram on the left illustrates the structure of your gut. You've got these little protrusions called villi, and on the microscopy image on the right, you can see those villi. They are the blue bits, and you can see that the upper bit of the slide will be the inside of your gut. And so for clarity, I'm going to call that the poop side, and your gut lining, the blue bit, I'm going to call the clean side, and everything below that. And so if you look at where these commensal bacteria are found, it's quite remarkable that you have this black area between the bacteria and the clean side, which I'm calling here the demilitarized zone. And so the poet Robert Frost wrote, "Good fences make good neighbors." And so good bugs respect barriers. And so these commensals stay in the lumen, in the poop side, and do not try to invade. Whereas pathogens that might arrive in the gut, for example, they will try to get into the clean side where lots of immune cells are present, and that can trigger a very powerful immune response. Now, this again is an oversimplification. It's not that our immune system completely ignores the presence of commensals. It's actually quite aware of them, and in fact, this equilibrium is not just maintained because they nicely respect the barriers. Those barriers are actively maintained, and part of the barrier is due to activities of the immune system. And the immune system is therefore needs to ensure that it is balancing these activatory signals that it's constantly receiving from commensal organisms, to making sure that it doesn't run out of control. And so we've been talking a lot about how you trigger things, but we got to remember that you can't just trigger things. You need to have a balance between accelerator signals and brake signals. And so all of the circuit is effectively under quite a lot of balances and checks, and you have immune brakes that operate at different parts, on different parts of this reaction that you see illustrated on this slide. And if you are unfortunate to have a dysregulation between the accelerator signals and the brake signals, and that could be because of genetic factors, or it could be because of environmental factors, then things can go seriously wrong. In the gut, you can get a disease called inflammatory bowel disease. And if you're interested in that, one of my colleagues, James Lee, has given a quick crash course on inflammatory bowel disease, which is available on the Crick YouTube channel. All right, so the last thing that I want to talk about today is cancer. Now, there are quite a number of cancer therapies, but one that has emerged as particularly important is cancer immunotherapy. And cancer immunotherapy effectively involves mobilizing the immune system to fight cancer. And in many cases, this involves the activity of those killer T cells that we have mentioned before. So I'm going to show you this video made by my colleague Gillian Griffiths in Cambridge, which has a killer T cell in red interacting with a tumor cell in blue. And there are subtitles on this video, so I will let you read them and just occasionally point out a few features. So one of the key features is that they have these poisonous proteins in red, which they inject into the cancer cell. And they do it in a very targeted fashion, making sure they're only going to the cancer cell, not to adjacent healthy cells. And quite a few killer T cells can gather around a cancer cell, and then when they perform their activity, there, poof, the blue cancer cell is killed and disappears. So this is quite an important mechanism that probably goes on very often in our body to eliminate tumoral cells before even they become a full-blown cancer. And one might argue that there is quite a lot of activity that prevents people from getting cancer, such that you wouldn't even know in principle that you had an incipient tumor. However, quite a number of cancers have caught on to this, and we talked already a little bit about how you need to have breaks in the immune system to make sure that things don't run out of control. Well, it turns out that tumors can co-opt some of these natural breaks to effectively put up a shield that prevents them from being killed by killer T cells. And this is what is called an immune checkpoint. It's effectively a break. And the suppression of T cell attack is an important clinical problem. Now, it prevents this activity of killer T cells, but immunologists and cancer biologists have developed what is called immune checkpoint blockade therapy. And what immune checkpoint blockade therapy does is that it effectively dissolves or takes away that shield and restores the killing activity of T cells. And so this is an extraordinarily powerful therapy if it works, meaning that some patients derive huge benefit. They can actually have their cancers cured by this therapy, and others don't derive so much benefit. And we're still trying to understand the rules, what it is that makes a patient a responder versus a non-responder to this type of therapy. And the answer is probably that there are multiple such checkpoints. So this is the break that we're trying to relieve with immune checkpoint blockade therapy. But as I've already alluded to, there are other breaks in the system. And it may be that from some patients, some of these other breaks are more important than the first one. And then the other point to consider is, much as we've discussed before, relieving the breaks is one thing, but we must make sure that we also have a mechanism for boosting the response, accelerating the response. And here we go back to this activity of dendritic cells, and a number of scientists, including ourselves, is interested in understanding whether we can actually develop drugs that specifically boost the ability of dendritic cells to show the cancer antigens to the T cells and prime the response. So here's just one little video from our lab that shows dendritic cells extracting antigens from dying cancer cells in order to boost the response to the cancer. And so the way that we envisage this is as sort of triangle that you see here. You've got dead tumor cells that can be phagocytosed by dendritic cells, as I just showed you in that video. These dendritic cells can now take those antigens to the T cells, activate the T cells, killer T cells can kill the tumors, and we can get this cycle going, and we can accelerate the cycle. Of course, conventional cancer therapies such as chemotherapy and radiotherapy induce tumor cell death. Immune checkpoint blockade therapy relieves the brakes on the killer T cells, as I've mentioned, and what I was just saying now is here's an area for future research where we hope to boost the activity of dendritic cells. So this is pretty much all I wanted to tell you about today, but I wanted to leave you with this important message. We really are studying the tip of the iceberg, and there's a huge amount of knowledge that we don't have yet. And the other important point to make is that some of these therapies, immune checkpoint blockade, for example, was not discovered by people setting out to make a new cancer drug. It was the product of very long and sustained investment into discovery research, into this research that aims at understanding the rules and the mechanisms by which the immune system works, the type of research that is very much the ethos of the CRIC, trying to understand how biology functions so that you can then use that information to design better therapies and better vaccines. So I think this is the tip of the iceberg, and if we explore it further, we might be able to build on our successes, build on a success against cancer, and make that even more powerful cancer immunotherapies, but also build on our success of, for example, eradicating smallpox through vaccination, and maybe one day eradicate other really important diseases that affect mankind today, such as HIV infection, tuberculosis, or malaria. So, thank you.